This method provides an efficient alternative to the conventional analog-based approach in obtaining amino acid overproducers.
This technique outcompetes the traditional analog-based method by providing accuracy, sensitivity, and high throughput simultaneously. This method sheds new light on understanding amino acid overproduction strengths and the translation of rare codons, and could theoretically be applied to all microorganisms.
This protocol relies mostly on basic molecular experimental operations, which are easy to follow, but may require multiple trials in order to achieve the suitable selection or screening stringencies.
A visual demonstration of this technique offers a direct appreciation on the performance of the selection and screening system in identifying amino acid producers.
To begin this procedure, add the vector and the marker fragments on ice in a molar ratio of 1:1 to 7.5 microliters of assembly mix to a total volume of 10 microliters. Incubate at 50 degrees Celsius for 1 hour. Transform 5 microliters of the assembly product into 50 microliters of component cells at 42 degrees Celsius for 30 seconds. Recover the cells in super-optimal broth with catabolite repression medium at 37 degrees Celsius for 1 hour. Then, plate them on LB agar medium and incubate overnight at 37 degrees Celsius. The next day, use a preferred commercial kit to isolate the plasmid. First, transform 50 microliters of the competent cells with 1 microliter of plasmid that carries the